They gave me 20 minutes to introduce you to the chief culprit of this obesity epidemic, which is the fat cell. Now, fat cells, we all love to hate them. Most of us have too many of these. The ones we have are frequently in all the wrong places, basically. So how do we go about sort of figuring out what's going on with these with these fat cells? I hear the bell swinging already, so it's like, we have to appreciate that fat cells are actually really, really important. They are my favorite cell type, and they really govern the entire sort of metabolic pattern within the system. So anything related to fat cells is going to affect metabolism. We see that here. It's going to affect all these organs. But perhaps we also need to be aware that there is quite a strong correlation between increased body mass index, obesity, and cancer incidence with a worse outcome for a lot of cancers across the board. I'm thinking of postmenopausal breast cancer, prostate cancer, colon cancer, pancreatic cancer. So there's a subtype of cancer types that are profoundly affected by obesity. And then last but not least, we don't give too much thought about infectious disease, but our fat cells get very old. So the fat cells we have today are probably on average 10 years old. So they don't turn over very often. So they offer actually a very welcoming, fertile ground for things to invade them because they're shielded from the immune system, hidden away, and they're in a candy store as far as energy is concerned. So particularly parasitic infections have been studied in fat for many years, but now more recently relevant will be COVID-19 related viral infections, basically, that can hit the fat tissue as well. So obviously, obesity is a seemingly simple, physical problem. It is a matter of a long-term positive energy balance. We have energy intake on the one side versus energy expenditure, and the difference is stored in our fat cells for the most part. So energy intake, of course, calorically dense foods, and on the energy expenditure side, we know that we all have a basic metabolic rate. We can lie in bed and we burn calories. Very few, but we do. And then, of course, as we are out there moving about, we have to stay warm. And of course, we move. So physical activity is definitely a contribution to the whole thing. How we handle potentially excess calories is all up to our fat cells. So if the fat cells handle that well, everything is fine. If the fat cells become dysfunctional, we actually have a problem, a very profound problem. So when you look at this, bathtub, where we want to be at the steady water level in terms of what's coming in and what's coming out. And it turns out that's a difficult task, it seems, because we are here tonight, because we're talking about obesity in that sense. And again, the fat cell is absolutely center stage. You know these numbers. We know we have an obesity epidemic out there. Whether that's Australia, you see the numbers here, or whether that's in the US, in Western Europe, it's worldwide, in fact, where we have this dramatic upwards trend towards increased BMIs across the population. And with that tracks very closely a number of different disease states. And I don't want to go through all the things that more obese individuals are subject to. Some of them here are listed, of course, some of the obvious ones. So when you have a dysfunctional fat tissue to an unhealthy expansion, it affects all these other organs. So the fat tissue is really center stage, whether that's neurodegenerative diseases, Alzheimer's, aging-related diseases, cardiovascular disease. We've heard about this. And of course, our closest correlate to the obesity epidemic is basically the diabetes epidemic. It goes hand in hand. The patterns overlay when you go countrywide in terms of the obesity incidence, the type 2 diabetes incidence completely overlapping in that sense. So there's clearly a strong relationship between the two. Now, the fat cell used to be actually a very simple cell. We didn't have too much respect for it. After we eat, as the sugar levels go up, the fat levels go up, the fat cell is supposed to store these excess calories. When we fast, it's supposed to release it in the form of free fatty acid. It keeps the brain going. It keeps the liver going in terms of the sugar production. But it turns out there is so much more to the fat cell than just sort of a classical fat blob, basically. They communicate. The fat cell communicates, and it communicates intensely. It's an endocrine cell, and it releases hormones. We call them adipokines because they come from the adipocyte, the fat cell, and they communicate. There are lipids that are being released that help communicate the fat cell, and then, of course, we have critical metabolites that are being made. And all of this has to be tightly coordinated, just like Maestro Ogawa here sort of directs his orchestra. All these adipokines, these fat-derived factors that interact with the various other organs in the system, they have to be coordinated in the fat cell, released, and our favorite one here is called adiponectin. It sort of makes everybody else go dance around in the system, basically. So this is a tightly orchestrated enterprise, and it can be easily dysregulated. So basically, what it happens is that we need to keep our fat cells happy, and we should not make them angry, because when the fat cell is angry, it really negatively impacts every other tissue. How can we keep fat cells happy? Of course. So fat cell is happy, everybody is happy, basically. And the fat cell is angry, everybody gets angry out there, basically, as well. So it is that communication axis between the fat cell and all the other organs that is so relevant, and that goes, a lot of things go so wrong as we expand our fat mass in this context. And each one of us here studies a different interaction between the fat cells, whether they are happy or angry, with another tissue. In our case, we love to study the crosstalk between the fat and the liver. The fat is dysregulated, the liver becomes fatty, and that's not always a good thing if you want to keep that mission of storing excess calories in your fat tissue and not in any other tissue. We understand quite a lot about the dysfunction of fat tissue. We know that basically our expanding fat mass is like a tumor mass, because an expanding tissue mass needs infrastructure. It needs vascularization, because we need to supply oxygen and the fatty acids and everything else to it. And that supply, as we expand, it turns out is probably not keeping up with the rapid expansion, and as a result, the fat tissue becomes hypoxic and undersupplied with oxygen, and then it starts to produce extracellular matrix fibrosis, so the fat tissue can become fibrotic, and as a result, it becomes highly dysfunctional. But at the same time, we also know the correlations between fat mass and, in the context of diabetes, insulin sensitivity. So for instance, when we are lean and healthy, when you plot this against fat mass at the bottom versus insulin sensitivity, or in this case, it's insulin resistance, actually. So with lean and healthy, we are quite insulin sensitive. So the resistance is low. Most of us are up there, the metabolically unhealthy obese, which has a resistance to the actions of insulin. But then there's an interesting subclass of individuals which are not all that infrequent, that are actually quite obese. They have quite severe forms of obesity, but everything is normal. They don't have diabetes. Their cardiovascular risk profile is low. They're perfectly fine. And we want to know more about how do these people do that. They have a higher chance of developing diabetes with age, but at the moment, they're just doing fine. And perhaps the most classical example as to why fat cells are important, we want them, we need them, are basically the individuals that we refer to as lipodystrophic individuals. So their problem is they do not have fat tissue, or very limited amounts of it. And that's a big problem, because they will become severely insulin resistant. Their liver will grow with overaccumulation of fats. So that's a classical case why we want that. Then also, not every fat pad that we have is the same. We know about apples and pear-shaped fat distributions, more female type fat distribution in the lower subcutaneous fat mass is basically beneficial. You retain insulin sensitivity, and then the male type obesity in the visceral obesity is of course closely associated with an increased cardiovascular risk. So we frequently say that with fat, it's just like with real estate. It's all about location, location, location. And that holds true across the board, basically. So it's very important to be able to basically push these excess calories into the right places, something which we haven't completely mastered yet at this point because we don't understand what determines the distribution of these excess calories. We are fascinated by this concept of metabolically healthy obese individuals. And again, they display, despite an elevated body mass index, a lot of normal characteristics. And we want to study that. And that's why we tend to go back and forth between the clinical examples, and we can use mice actually to genetically manipulate them and study cause and effect much better than what we can learn clinically, which are mostly correlations, which of course matter, but in the mouse we can actually test this very specifically. So for each one of these states, we have mouse models available in which we've modified, tweaked things a little bit. We have lean, healthy mice. We have obese, unhealthy mice. We have very lean mice, which are lipodystrophic. They don't have a lot of fat, so those mice are in trouble as well. And then, of course, we have the healthy, obese mouse as well. And that's what we've heard about initially. Those are the world record mice, basically, which are very, very obese, but at the same time completely retain their insulin sensitivity and a healthy cardiovascular profile. So something has gone right here in the sense that we allow the fat pad, in this particular case, it's the subcutaneous fat again, that expands to a very, very large extent in this case, to the point where the mice can no longer really walk around very easily, and that's when they start to reduce their food intake because they can't get to it anymore so easily. But other than that, they're actually perfectly fine and happy mice. We also want to bear in mind that there's more than one type of fat that we all walk around with. What we are thinking of when we talk about the obesity epidemic is basically the white fat. This is the classical fat pads that we're all having. Whether that's a visceral fat pad or whether that's a subcutaneous fat pad, they're white adipocytes. Now, there's a different type of adipocyte that is not focused on storing energy, but actually focused on burning energy. Those are the fat cells that keep us warm. And we refer to them as, they have a couple of different names, but they're basically brown adipocytes because they're brown. Their color is, they're full of mitochondria, these little energy burning intracellular structures, and they create heat. And for the longest time, there's a different flavor of them, beige adipocytes as well. They can pop up in our subcutaneous fat tissue, or they can pop up in specific places in the system. And we can visualize that actually relatively easily when we use an injection of radioactive glucose in this particular case. This is a classical technique that's used, of course, in the cancer clinic to diagnose tumors, because tumors will have a very avid tendency to take up glucose. But incidentally, somebody noticed, and this is all quite recent, in the last 5 to 8 years, they finally sort of realized that when you look at these scans, basically, that there was a seasonal difference, that there was a lot more signal in the winter than in the summer. And it turns out the textbook used to say that only babies have brown adipose tissues in humans, because as they come out, they of course have to stay warm and they lose that tissue very quickly. However, as adults, we can actually also have brown adipose tissue, and we can induce it in the cold, and some of us are better at that than others. But that's certainly one mechanism by which we can spend excess calories by just essentially keeping warm. That's why my office temperature, I like to keep it around 14 degrees centigrade, because it does two things. It will, on the one hand, burn excess calories. This is not just God-given here, so this. And on the other hand, it keeps conversations extremely short. So 5 minutes, we're done, moving on, basically. So how do we control our weight? That's obviously a very important feature. The accuracy, despite the obesity epidemic, the accuracy by which we maintain our weight is quite remarkable. Now, someone like me, I probably consume close to 1 million calories every year. And if I, at the end of the year, God forbid, have added 1 pound to my weight, that would be about the net, 4,000 calories in that pound of fat, basically. And then you do the math on the back of the envelope and basically realize that despite gaining 1 pound now, this is incredibly accurate, right? It's with less than 1% accuracy. And when you really do the math in terms of what that means for your daily caloric consumption, it turns out it's basically about 10 or 11 calories a day that you will be off. So that's a tic-tac a day, right? That there's nothing. Nobody goes through life counting calories like that. So it tells you that the process is regulated, tightly regulated, because we don't watch what we eat, we just consume. And at the end of the year, when we are lucky, we come out with the same weight, or maybe we don't. Maybe we're off by a pound. That's bad news when you're off by a pound over the next 20 years. That means you actually gain 20 pounds extra. But it's incredibly accurate. Now, you can actually, some of these things can be done both with humans in the clinic, or you can do it again with mice. You can see, for instance, here, we've calorically, this paper is not our work, but it's what we all see in the lab. We've calorically restricted the mice. We split them in half. One half gets limited calories, the other one just keeps eating ad lib, and they of course gain weight, the ones that eat ad lib. And then at substage here, you can see we kind of stopped the food restriction and gave them all they wanted to eat again. And look what happened. Immediately, the weight goes back up. And what's most curious is that it doesn't necessarily overshoot or go somewhere else or stop halfway up. What you see, it goes exactly back to where the control mice were at the same time. Tightly regulated. What is that switch that we all carry in us that will drive our weight, even though we go on a diet repeatedly, we always drive back to our maximal weight that we previously had. And we still don't have the full answer to that. So if this is so tightly regulated, why do we get obese? Well, again, it's a complex set of regulatory events, but we can do this with a mouse. We either give the mouse the choice of having a chow diet, which is much more sort of a robust low, nicely balanced diet, basically, or we give them a high-fat diet. And you can see that the mice on the high-fat diet gain more weight. So it's kind of the perfect reflection of what we see out here in the wild with us, that basically excess caloric exposure leads to increase weight. But then again, it's not just the environment, the availability of calories. There's a genetic component in there as well, and that's an important phenomenon as well. We have, in this case, a black mouse. We call them the C57 black 6 mice. And then there's the FVB mouse, which is a white mouse. The fur is white. And you can see that they both get exposed to the same diet. We all work with the black mice because they gain weight and their fat expands and they become diabetic, whereas the FVB mouse gives us a much harder time when we want to gain have them put some weight on. So that just shows the genetics of closely related species between these two mice matters a great deal. And of course, that's the heterogeneity that we observe in the human population as well. We all come with different set points and different genes, basically. So the one thing we have to bear in mind for the discussion is, yes, there is a set point for each one of us. That set point seems to be different. We don't understand when and how that set point is being given, basically, but it's clearly a combination of genes and environment, multifactorial. Yes, there are examples of specific mutations. A protein is mutated, it causes obesity. That exists, but for the vast majority of us, it's much more complicated than that. So there's an interplay between genetic and environmental factors, and then, of course, there is the pharmacological intervention out there as well. So I guess there is hope at this moment. We've come a long way. to be able to actually intervene pharmacologically and bring the weight down without an excess number of side effects. So this, for instance, you can see, this is now a clinical study. Those are months, so this is a 2-year study. And you see here, these people have been counseled to change their lifestyle. They will exercise more, but basically they stay the same. And this is now a pharmacological intervention which acts on the, what we call the GLP-1 receptor axis, that lowers the body weight and keeps the body weight down here. And this new generation of drugs now, these GLP-1 receptor agonists that are being developed and are being approved left and right in various forms now, they actually turn out to be very effective. And unlike previous weight loss drugs, which were just packed with side effects, where you had to be concerned about depression, you had to be concerned about cardiovascular side effects, these seem to be actually safe. And they keep the weight on for as long as you take the drug. That's the other part again, of course. So when you stop taking the drug, what you see both in the mouse as well as clinically, the weight pops right back up to the original weight. No matter how hard most people basically will end up in the same place that they were to start out with. So basically, there is hope. I think we can talk about interventions that will give you 20% weight loss realistically, not as an exception. The average weight loss on some of these drugs is in the order of 20%. Doesn't sound like that much, but remember, you walk around with 250 pounds body weight, 20%, you're losing 50 pounds with relative ease. So that's a very significant improvement in your weight. And with that, we have improvements in cardiovascular outcome. Presumably, we have a reduction in the cancer risk. The diabetes is reduced. They may not completely go away. A lot of issues are being solved with a 20% weight loss. Not everything is solved, but a lot of things can be solved on that. So for us, of course, we need to find out more about how this whole thing works, who is really in charge, basically. And we like to argue about this. Now, it's clear the brain is important. The brain is the executor of all these commands and will pull all the strings. But who does the brain listen to? Now, someone like me will say, well, they'll listen to the fat cell, basically. It's the fat cell that communicates from the periphery up how much fat we have. What do we need to do about that? And then, of course, the brain will respond accordingly in terms of perhaps reducing food intake, increasing energy expenditure. We do need to do more research, because you also have to realize for every dollar, that's the same in Australia as it is in the US, for every dollar that we spend trying to fix the problem of a type 2 diabetic or cardiovascular disease phenomenon, for every dollar spent in the clinic, we spend less than 1 cent on actually trying to figure out what's really going on and how we could fundamentally change that process rather than just putting a Band-Aid on and trying to fix things at a very late stage. So with that, I want to thank you for your attention, and I think we can move on to the next stage. Thank you.